Welcome to another session of the International Crimes Investigative Committee. Today we're going to be talking about a topic that has been on many people's minds, but many people have discarded it as being somewhat wacky. We will try and see what is really going on with these lipid nanoparticles, what is in the vials, how does the stuff that's in there, whatever is in there, interact with outside forces, maybe microwaves, maybe 5G, we don't know. But we're going to try and zero in and see if there is any truth to the rumors that we have heard, because what we're talking about is not just corona, we're talking about the entire agenda of the Great Reset, which includes population control, and that includes population reduction and transhumanism. That in itself is wacky enough, I must say. We will try to not sensationalize anything, but we will not downplay anything either. We'll try and look at everything in the most rational manner possible. We have a great panel of experts with us. Dr. Mike Yeadon is, for the first time, our co-host in this. He will be able to ask the right questions. I'm just an attorney. I need experts to explain everything to me. Dr. Mike Yeadon is the former vice president of Pfizer, and I think, Mike, you've been their chief science officer for 16 years, if I'm not completely mistaken.
Yes, I was, for allergy and respiratory rather than the whole company, yes.
Mm-hmm. And I'm gonna, please go ahead, Mike, and introduce yourself, because your biography is so impressive that I don't have all the details.
Well, I don't know about that, but I always say to people that I'm doing my normal job in extraordinary setting. That's all. My job always has been to try and understand, what's going on in health and disease. What can we possibly do to intervene in a way that would bring benefits of value to a patient without unacceptable side effects, because that's always a problem. It's 100 times easier to hurt people than it is to produce a benefit. So, in fact, the default expectation when you mess with biology is that you break things. That's the truth. So, people may know I was at Pfizer. I'm a PhD pharmacologist. I was a biochemist, toxicologist, original training. 32 years in biopharmaceutical R&D, 10 years of the last 10 years of which I was in biotech. I both consulted to biotech, about 30 companies, and also ran my own biotech, and was very lucky to get acquired by Novartis in 2017. So I was comfortably retired when this crime began in front of me, and within a few weeks I transformed from a normie who listens to the national media and thinks it's pretty much correct. I'm just going to have to come back on the call in a moment. I'm just receiving an urgent call. Okay? I'll be right back.
This is, by the way, exactly what happened to me. I would consider myself to have been a normie, and then Corona struck, and all of a sudden, nothing seemed to make sense anymore. We will start with Dr. David Nixon, who is a scientist are you from Australia, or do you just live in Australia, David?
Good morning, good afternoon. I'm living in Brisbane, Australia. I'm a family physician, so I'm a general practitioner, as we call them over here. So I've been spending the last 25 years managing family patients of all ages and all conditions. In about June of this year, I bought myself a dark field microscope, and I started looking at patient blood and the vaccines under the microscope. So I'm keen to talk through what I've been finding with respect to the Pfizer vaccine in particular.
And you kind of gained notoriety because of the findings you made through the dark field microscope when you appeared on the Maria Z show. And that is what we're going to start talking about in a minute. We also have Shimon Yanowitz, who is, you're an engineer from Israel. Am I correct when I say you're an electrical engineer?
Yes. Hello, Reiner. Yes, I'm from electrical
Shimon.
engineering. You're correct.
And we also have Maria Mihalcea. And please go ahead, Anna. You have a very unusual background. You come from Romania. You grew up for a while, at least, in Germany, and then you went to the United States.
Yes, so I'm a board-certified internal medicine physician in the United States. I have a PhD in pathology. I have my own anti-aging clinic. I've been active in the COVID resistance for over 2 years. And then I got involved with this international collaborative effort to investigate the C19 vials, and have been collaborating with Shimon and David and others. I've also explored the effects of vaccine shedding through markers like D-dimer, and I'm an advocate for new ways of screening vaccinated individuals to prevent sudden death by looking at all aspects of what could be in the vials and what could be affecting people, including this type of strange and weird technology that we have found.
Thank you. And we have Karen Kingston. She needs really no introduction, and we've spoken with her quite a few times already, but this is important because she has been, for a long time, she has been taking a deep dive into what is in the Pfizer vials. And once upon a time, if I'm not completely mistaken, Karen, you even worked for Pfizer, correct?
I worked for Pfizer a long time ago. I mean, out of college, I was in sales, and then I moved to their agency side. They moved me into marketing. So I did work for their agency. I worked on some brands you've probably heard of, like Viagra and Lipitor and products like that. But I'm a strategist and an analyst. I ran my own agency for several years. Pfizer was a client of my agency. It's called Veritage, which means truth. I was very good at what I did. I was not well liked back then either. I was hired because a lot of times companies would be in a foxhole because their marketing agency and sales team would go rogue, and so I would have to take them back to what they legally can say and what was submitted to the FDA. So, and my expertise is also to go through hundreds of pages of complex clinical data and simplify it so that physicians can understand it, and then simplify it again so that consumers can understand it. I did not do this on my own. I had a number of PhDs and MBAs, and I also had designers and editors working for me. So I'm doing this on my own now, but hopefully we can get a unified team together to move forward to really get the truth out there.
That's exactly what it looks like right now. Well, let's start off with you, David. What is it? Because your findings kind of made a splash within the resistance, if I may call it that, when you were on the Maria Z show. So what is it that you found? Right.
So this, just quickly, just a little bit of background about how I got involved with this. This is a quick short video of how the Pfizer vaccine appears under a dark field microscope at 100 times magnification. We see this sparkling background here, and then often we see these abnormal structures which appear to amalgamate together. And these have been described as being cholesterol shards, nothing to look at, but there's been concern about their nature certainly from a number of groups internationally. This is a photo-stitched image of a couple of drops of Pfizer vaccine that have been left overnight, and as you can see around the periphery, there are some strange crystal growths, or some strange structures that appear to grow in the nature of a crystal. Some of them are really quite complex in appearance, and I've certainly showed these images quite widely. some of the images do not look like naturally occurring crystals. They have internal structures that appear to be different from what one expects from a naturally occurring crystal, and there's often associated debris appearing beside them. One thing I did early on was I put a slide inside a Faraday bag, and I discovered that when I did that, the crystals, the structure wouldn't grow. All I got was these black dots. But when I didn't shield the slide, I got these structures would appear the next morning. So what happened next was that I revisited one of the slides that I had done previously 10 weeks before, and discovered that the crystal-like structures had actually changed in their appearance, and they were present not only on the periphery but all through the sample. And when I did a close-up of these structures, they were fundamentally different from the original ones. And they had, the appearance of a much more concerning nature. So I've got a few examples of these. And these structures were often associated with these ribbons that were present as well. So I've just got a few examples of these structures and the nature of their design. And as I said, they're often associated with these ribbons. And some of them are really quite concerning in appearance. And they're often associated with these ribbon-like structures, which weren't present themselves 10 weeks previously. Mm-hmm. And we can also see that these smaller structures in the background appear to be a smaller version of the larger one. So if I zoom in on this smaller one, it'll have a similar appearance to the larger structure. And then what I discovered overnight, all of these structures disappeared. That is all of them except for a couple, and I focused on the ones that remained. And this is a photo of the night of the morning after all the structures had disappeared. The following, or about 6 hours later, this was all that was left of that crystal, was the central structure. And then over time, all of these structures that were visible in this field, this is 40 times magnification, just slowly disappeared. And at normal speed, it's very difficult to detect anything happening. But when I speed up the video, this is an hour recording, it's possible to see the structure slowly disappearing. So this is another clip of the process. This is the central crystal that I showed in the previous photo. This is now 200 times magnification. And at normal speed, there's the appearance that nothing's happening. But when the video is sped up, it's possible to see that there is in actual fact quite a lot happening in the background and to the square itself. So I'd just like to share a second video I've got of this process, which I need to share as a separate file. So I'll just stop that one and start this one. So this is a similar example of this process, which I'm calling micro deconstruction. and at normal speed, nothing is appearing to happen. But this video here is almost an hour. In real time, nothing seems to be happening. But again, if I speed it up, we can see that there's quite a process going on. This structure on the left here disappears first, and then the central crystal just disappears. And this material around the central square, which was inactive to start with, certainly seems to be involved in this process. So I've got quite a few examples of this deconstruction process happening, but it was the video of the construction process itself that was the one that I discussed with Maria, and I'll just forward to that now.
David, this is Mike. I'm looking with interest, and I may ask questions. I'd like you to, receive it as an enthusiastic skeptic. That's the way
Yep,
I've always been. I've always been about science. I'm always enthusiastic, but I always think I'm not sure about things. So when I ask, that's the approach. It's not that I've a concern.
No.
But so when I look at the film where you showed the disappearance of the large square, what occurred to me, and I've had very little exposure to microscopes, just enough to be dangerous. Okay, so that what it seemed to me to be possible was that the object was moving out of the focal plane over time, because I remember playing badly with say smears of cells from an airway, and I would sometimes realize if I pressed on the stage that the slide is mounted on, or just move the focus slightly, sometimes an object can appear and disappear exactly as you described, as you showed it. And I'm thinking, what steps did you take to convince yourself, as well as other people, that you remained focused on the same plane all the way out, all the way through, so as to be able to reject that possibility? I think you know what I'm saying, that what I've said is true, that yes, objects can appear, disappear and appear if you change the focal plane by a few microns.
Yeah, I quite agree. But the video was shot over an hour, so an hour, and so it didn't appear and disappear, by touching the slide.
No,
because it happened so slowly. This is the next video, and this is being sped up about 400 times. And in real time, you don't see anything happening at all.
So my reaction to this as a biologist who has a little bit of physical chemistry is my first reaction would be there's some evaporation of some solvent, which over time is leading to really small changes, but those changes are large enough to register as movement. It's possible that it's
Mike, may I just ask you, just from the perspective of what is this structure doing in a vaccine vial?
Oh, yeah, it shouldn't be in there. So I will say, well, let me just take a couple of steps back. These vaccines should not exist at all. Mm-hmm. As a biologist, I can tell you it's literally impossible. If the narrative of a new virus had been true, which I don't believe, but if the narrative of a new virus had been true, I can promise you that generating an intramuscular vaccine was an inappropriate strategy that could never have worked. And the regulators and the drug companies have the data to prove it. And in brief, we know 10 years of intramuscular flu vaccines did not reduce incidence of hospitalization or deaths from influenza-like illnesses. So we already know that the elderly, vulnerable people do not benefit from vaccines, almost certainly because their immune systems are senescent.
So Mike, we are watching this very interesting thing, and I understand that you have your opinions, but maybe we can allow the presentation of David just to kind of explain what he's
I was going to get to you. I was going to get to your side by saying that vaccines are an irrational strategy, so they shouldn't even exist. The fact that they exist is fraud. The fact that they exist and have been jabbed into billions of people is a deliberate crime. Okay, so I'm with you a long way. I just don't know what I'm seeing. That's all.
Yeah, I think it's important to ask the right questions, and I think that was a good question to ask. Okay, let's go ahead.
Yeah, so I've just got this on pretty much on repeat and focusing on the different areas. I think that's the end of that. This is the same structure 48 hours later, and these are the structures that were moving earlier. And what this video is just showing is the increase in complexity in terms of the lights, but also over time I've captured changes in the appearance. So there's lights that come on and off on this structure. That one went off. That's not quite so obvious. Yeah, so down the bottom, this light appeared, and this structure moves.
David, do you have any idea what this might be?
No. I mean, it appears to have intelligent design, and it seems to be acting in a way that would suggest that it's electronic, but that's only a guess, obviously.
I remember something else from your presentation on the Maria Z show, and that is that you said that this movement stops when you put the slide into was it a Faraday cage?
Yeah, so that was the part of the presentation that I quickly showed at the beginning. When I put the slide inside this bag which shielded it from the wireless router, I didn't produce the structures. When the slide was on my microscope, these structures would predictably appear the next day. So the shielding produced an effect of stopping the structures from appearing.
Which seems to indicate that it's not self-assembling, but some outside force may be having an influence on whatever kind of assembling is occurring here.
Great.
Reiner, I would say that this is still counts as self-assembly. Only the self-assembly process requires some energy source. So this could be either thermal energy, maybe just the injection into the body where the body temperature is elevated with respect to the ambient temperature. And there could also be contribution from an electromagnetic radiation source that could also flare up the dynamics of this process.
Yeah, the definition of the Tesla-phoresis process is self-assembling, but it is initiated by an outside signal.
So David, again, forgive me, just for rigor, when you put a slide in the bag, did you remove the slide from the microscope so you could close the bag, or is the bag still open? That's one question. And the other one is, when you put it in the bag, you change more than just exposure to EMF. You may exclude or reduce light. You may exclude or change temperature or air currents. And so I'm pointing out that although you may attribute the change to blocking EMF, you would need to do more experiments to be sure that that's what it was.
Yes. I did run the experiment overnight, so there was no light on either slide.
Okay.
But
great.
The slide was in a slide case, so obviously there was less oxygen available, and the humidity would have been different, and the temperature would have been different. But I couldn't control those things.
No, I understand. Just wants to make the point there.
I did run another trial where I put a slide on the router on my microscope and in the bag, and that particular night the router malfunctioned and was turned off all night, and I didn't grow anything on any of the 3 slides. So that was interesting.
Yeah.
But yes, it's a very much a non-scientific experiment.
No, you're in a position where you're in scientific discovery. So you've made observations, and they could be exactly as Shimon mentioned. It could be it's responding to an external power supply, but maybe not. And so it would be incumbent, to try. I always try and break things. I would think, okay, if I'm right, then if I do A, B, and C, then this would go away, and if I'm wrong, it won't. And so we have to be our own best critics if we want neutral people, and especially skeptical people, to take anything seriously.
Absolutely. So one of the reasons I tried this was after seeing Matt Taylor, electrical engineer who's in Ecuador, demonstrate a similar finding with respect to wireless router. You've spoken to Matt recently, Anna. Do you want to talk to that?
Yes, absolutely. So he has found a very similar finding that basically showed that that he was documenting self-assembly, and he also did a time-lapse video of that, and so this was clearly showing some form of circuitry. And Matt is an electrical engineer, and he clearly said, it's like, look, you cannot say that this is a cholesterol crystal, salt crystal. This is what a lot of the critics are saying. And I just want to mention that, first of all, for example, in the United States, it is a criminal offense to look at the Pfizer vials. So every single person who has done this research has done it undercover and under extreme danger for themselves. And so the people who are asking for rigorousness, which would be university setting type of scientific elaboration, we cannot provide this. We are people who are endeavoring to help humanity by bringing this information forward. And it should be the pharmaceutical company, the FDA, and the CDC who disprove us and really show that they are doing the studies to say, what is this? Because we, in the case that we're in, are not able or don't even have the means to be able to do this with a scientific rigor that would be acceptable to the scientists who are denying this possibility.
No, I take your point entirely. I agree that there are clearly materials in these vials that should not be there. There's no question. These are, I would say, without question. I've worked quite a bit with Hedley Rees and Sasha Latipova, and that together their experience and reading documents, apart from anything else, and knowledge of due process says it's not possible that the industry has manufactured what it claims to have manufactured. So I'm not that surprised when you look into some samples and find it's not as described. Basically, I mean, and Reiner will know, I've gone very far for someone who was a normie and in the industry, that the whole thing is a crime. Not, as I say, these materials are an inappropriate intervention in human beings, even if the story of a new virus was true, which I don't think is true. So I am with you. I am more than suspicious. I am certain these materials have malign intent, right? What I don't know is whether this is evidence. I wonder, this could be evidence of reckless, slapdash, casual manufacturing by groups that are not qualified to make pharmaceutical grade anything. That's one possibility. The other possibility is there is some, deep state CIA technology in here. And it could be either. Honestly, it could be either. But I don't need evidence. I've already got evidence of severe crimes. So if I'm going to be persuaded that it's an additional crime, I will have a high standard because I'm already over the line on the principle.
Mike,
These things are toxic.
Can I please interject?
Can I address this?
Sorry, I want to say something about the work of Matt Taylor that Anna is showing now. So let me just give credit to our colleague, Matt Taylor. He's also he's an electrical engineer, and I'm also a scientist from electrical engineering background, but also a lot of background in microscopy and microelectronic fabrication. So when you look at this thing from the point of view of an electrical engineer, you have nothing but to assume that this is an electronic circuit that is self-assembling right before your eyes. And as David shows, it's not so quick. It could take a day or two or three or a month, depending on how much energy is supplied to this. So let me just say that David and Matt Taylor's work are in corroboration with each other. And Reiner, regarding your previous, Mike, regarding your previous question of things going in and out of focus, I will want to maybe help David by saying that David has used an extremely low magnification in this microscopy. So that means the depth of field is rather large. It was nothing like something like a 100 times objective. It was a 20 times objective, which is not a very high magnification.
Agreed.
And the depth of field is rather high, and we have the same exact phenomena found by Matt Taylor, not using dark field. He was using bright field. It looks much more impressive on David's very advanced dark field microscope. And I also want to say that on my side, I found, in addition to what seems like electronics, I found other structures self-assembling. I will be able to show them later. So this is not just the materials and the excipients that are in those vials. It's this amazingly advanced technology that seems to make new things out of these materials and excipients. And as David has shown, not only can it grow structures, it seems to be able to eliminate them thereafter, thereby maybe getting rid of some evidence. This is, I'm not making up these things. You can see for yourselves, right?
I was wondering, David, have there been any reactions from the mainstream? Have there been any statements saying, for example, oh, you're imagining things. These are just crystalline structures dissolving or something like that, or nothing?
Nothing. Crickets.
Huh.
That's not true. So Ryan Cole said that all we were seeing was cholesterol, salt, and sucrose crystals. And that there is no nanotechnology in there. We're not saying it's nanotechnology, it's microscopic scale, but a lot of people are saying that this is just normal. But, cholesterol crystals do not look that elaborate, neither does salt or sucrose. I discussed this with Sasha Latipova, and we showed multiple, comparison structures. So I think that If you look at some very, very advanced images, even from Life of the Blood in New Zealand, I mean, this looks like microcircuitry, and any engineer that we've showed this to said, this is a circuit.
Mm-hmm. What is her take? What is Sasha's take on this?
Sasha is extremely concerned about all of the metals that are in there. She's concerned about all of these structures, the ribbons. She's saying, what is this? She's published on Trial Site News about David's finding. She is actually leading our effort at the International Research Collaboration. She's extremely concerned about this and wants answers, as do we.
Mm-hmm.
I just want to make a comment. So, I mean, I was attacked by Reuters last year when I said that these contain devices with graphene oxide in the Associated Press. So after I went on Stu Peters in July of last year. So you can find those on my Substack. But for what, I talked to attorneys last year when the data showed up in VAERS. So as far as finding, there's a couple approaches to discover what's in these vials. I said under any other circumstance, even if it was a vaccine, if you had that VAERS data, what you would do, and this is why people started looking at the lots, what you would do is what Johnson & Johnson did when they did a recall. You would take a look at the deaths, you would look at the lot numbers, and then you would recall those lots, and you would inspect them with an independent lab because you wouldn't be able to explain why there's so many deaths. So that's when people started saying, look at your lot. They didn't do the next logical legal step, which is then to recall those lots because they caused severe disease, injury, and death. And then an independent lab under a court of law would then look at those lots. So the lots that are causing the deaths in VAERS in and of themselves should be recalled and looked at. And now that the FDA approval has happened, later I'll share my screen. If you take a look at what they approved as far as the ingredients, the manufacturing processes, and the toxicity, 80% of it's redacted. Under the FDA law, since it was approved, we have the right under FOIA to know what those ingredients are. These are not biologics, though. We're not going to get it from CBER. We're going to get it from CDRH. How do we know that? Because pegylated lipids are classified as devices under CDRH. There's a logical explanation, Reiner, but for some reason, the logic and evidence I present is called not credible and ignored. And I do med legal review processes for companies. I've set them up for large Fortune 500 companies before they were Fortune 500 companies. So there's processes, there's logic, and I appreciate all this information. Obviously, I've always been supportive of researchers and the scientists, but I'm looking for a methodology that would meet the critical needs of everyone in this movement, including Dr. Yeadon is talking about, and as far as them saying this could have been a slap-happy hazard manufacturing mix-up. No, it's not, because the ingredients are redacted. So any expert who's saying they're 100% confident there's no nanotechnology or devices or graphene oxide in here, how do they know when 80% of the ingredients are redacted? That's the first thing. Second of all, there's over 60 manufacturers. Mike, let me finish. There's over 60 manufacturers, including GlaxoSmithKline, Pfizer. I'm trying to think of. There's 60 global manufacturers of these lipid nanoparticles. Did you guys know that? I mean, it's published. There's a $12,000 report you can buy. It's on my Substack. So this is not a mistake when there's over 60 manufacturers globally, many of which are household names. That are manufacturing. The Sanofi is one of the manufacturers of the lipid nanoparticles. Why is that?
Mike wanted to interject.
Sorry, don't get me wrong. Although it is possible, because the vaccines have no purpose as vaccines. Okay, immunologically, you can't rescue elderly, frail people with vaccines, right? So the whole lot should be withdrawn and flushed. Now, so it is a fraud in itself that they even exist, and they're lying to the public and the doctors and the FDA and you every day when they tell you come and get you. It cannot possibly work. The evidence, I'm not just guessing from principle, I'm showing from decades of use of intramuscular vaccines against influenza-like illnesses. They don't work. So the whole thing is a fraud. So don't get me wrong, I'm definitely on the side of this is a crime. The question, I think, is only what is the crime?
Yeah.
So my initial proposition was they are expressing this biologically poisonous material called spike protein. but I'm not sure that's an adequate explanation. You talked about the variation between lots. That seems to be a possibility, but that has not been explored. So I don't know whether the variation has got anything to do with the amount of spike protein expression. It could easily be to do with deliberate malfeasance. Maybe something has been added to it. So the whole thing, we don't need to just withdraw some lots and have a look and see if they've been made right or not, or something been added. They should all be withdrawn and thrown away. So
I agree.
Companies around the world are conspiring and lying and providing a false narrative to inject materials of unknown provenance and purpose into the entire global population. So that's pretty wacky, what I've just said, but that's what I believe. I could stand up in front of a court and say so and justify it. So it's just a question of what are they doing? What are they doing?
We could have started by recalling the lots that caused the deaths and VAERS.
Absolutely.
Legally,
Absolutely.
that could have
That'd be a good
happened.
start.
It still could happen. I'm just saying that there's methodologies. I understand there's processes to get legally and intellectually and socially to get to a point. I'm just trying to express that process too, as well as as well as
but
support
again, because they've broken all the laws. I think, honestly, 3 years ago, I would have called myself crazy for saying what I'm about to say, but it seems to me every institution and structure that would normally serve to protect, the public has been suborned, and they're not going to respond to anything you do. Mm-hmm. That's my concern. Now, obviously, everybody should try their hardest, but as a scientist, and looking at what's happened, I would not anticipate we will get success because everything is broken. That's sad to say.
Can I add to that, Mike? So let me just tell the audience that I've known Reiner and Mike for I think at least 2 years now.
Yeah.
We've met on the meetings of Children's Health Defense 2 years ago.
PCR test group, yeah.
2 years ago, before these injectables, I call these injectables, Mike, I refuse to call them vaccines.
I agree.
Before they were ready, I gave their a presentation on nanotechnology that I anticipated then would be in the later introduced injectables.
Yes, I remember.
And it was very hard at the time to convince people, even myself and certainly Mike and Reiner, that I wasn't, confusing science and science fiction.
Yes.
Fast forward 2 years later, Mike, and I have yet to see one demonstration of any spike protein pulled out from the blood of a single person. They don't have anything like that to show. Further than that, I have yet to see one electromicrograph image of a lipid nanoparticle.
Exactly.
I can show you.
The whole thing could be just a narrative. The whole thing could be a lie.
Exactly.
I've said to people, how many times is the right number for public health officials to lie to you in a way that could cost you your life? It's a rhetorical question. The answer is 0. Well, I'm confident that I can prove that maybe 5 or 6 major narrative points that come from everywhere, from Fauci, the companies, the regulators, are lies. And so why would I trust a single word they say about anything else? And the answer is I shouldn't. So I agree with you. Although they tell us that the sequence causes expression of spike protein, it could just be a story. I'm uninclined to believe anything they tell me anymore.
Mike, this is why we are looking through microscopes at these things. And what we are finding when we are looking through these microscopes are amazing things that are in complete contradiction to the narrative. Furthermore, no one else has come up with any microscope findings that supports the official narrative. Now, it seems that like we are in some kind of genocidal war. So we are facing an enemy. Why would we believe the narrative that the enemy is telling us? It's complete nonsense.
I don't personally. So I agree with you. That's why, what they have said about a virus and the need for a vaccine is definitely wrong. It's demonstrably wrong. So, yeah, I've said to people, I don't know what's in the vaccines. I do not believe that they have been demonstrated to be safe. I don't think there's any useful function to be served. So please don't get any injections and stop your children being injected. And join me in pointing out that officials are lying to everybody. But
So
you can
Mike,
tell, that's as far as I've got.
Mike, I would like to interject here. So one of the reasons why it is important to look at this information is that it's been clearly shown within our groups that the mRNA is either absent in the vials or there's only RNA fragments, which is if there is no RNA, then there is no spike protein in many people. And then the second thing is that metallic, they call it contaminations have been found. Things like cesium, barium, gadolinium, aluminium that are extremely toxic on their own, particularly if they're nano size. And then the next thing is that possibly graphene cannot be ruled out because if there's carbon and oxygen, then you cannot rule out a monatomic layer of graphene that is potentially self-assembling. And the regulatory fraud that's been shown is just phenomenal. Why is this important if we don't have any, legal ways of stopping these pharmaceutical companies or however long it takes? Because 5.3 billion people have been injected with this. And the current vax protocols do not include detoxifications of metals. They do not take into account graphene. And if there are things like EDTA, like humic fulvic acid, like nitric oxide, that nobody in the narrative of the frontline doctors is even discussing, could be helpful to 5.3 billion people, then our conversation is worthwhile.
Everyone agrees.
All of what you said is true. None of the materials you listed have a therapeutic or acceptable purpose, and as you say, I don't believe any official description of why they should be in there, or if inappropriate, what they've done about it has ever appeared. So the whole thing is a dreadful calumny, a crime. And so I have approached it logically, and I think even with what I know already, I think I can prove to an adequate level of necessary proof that what is being done is completely inappropriate and therefore dangerous. There may be even more danger lurking in it. I don't have the right skill sets to understand what to do with the fragmentary information that I'm seeing. It's very troubling. But I know that I don't know enough to know what to do. Okay, so, and I accept the point I think Anna made that, we don't have MIT labs and 60 postdocs to go and do all of the experiments necessary. We are where we are, and it's troubling. But I stick with what I know already because it's adequate description of a crime. But I agree that there are definitely additional materials that you look down the microscope and it's very worrying. I can't go as far as to say that's a self-assembling nanoelectronics that might have these properties, because that might be true, but I just don't think we've got the evidence for it. I encourage
Mike,
further.
Can you just allow the presenters to continue with their presentation before you come to the final conclusion?
Hold it, Anna.
Fine.
I think this is the right approach. Mike is not saying this is wrong. He's just saying we don't know enough to come to any conclusions. Because if this is a crime, and I think we all agree on this, the big question is, what kind of crime is it? And in order to answer this question, we need to know what is it that we're finding and what purpose do the things that we're finding serve. We know that some of the heavy metals that have been found in some of the vials are, of course, toxic. But what is it that David has found through his dark field microscopy? Is there anything that, of course, we're speculating, but this is probably how you start any scientific research, because you have to start to think about what the goal is that you want to achieve. What is the result that you need? Me as a lawyer, I need to know what this is and what it does. Do we have anything that gives us an answer to these questions?
Yeah, I mean, we do. I mean, so what they're showing, what the scientists and researchers and doctors and Anna are showing under the microscope can match up to what is in patents and then in peer-reviewed journals. Again, it just takes time for me to do this. I've done it before. I did it with Carrie Madej last year, with what she found, and Dr. Zandra Batra, and it's not a coincidence with what they're seeing underneath the microscope. It looks exactly what's in the patents. I even showed the magnets that were used as drug delivery devices that attach to the bone in the body. So these are things that were overlooked last year, and I can do that again. But I think from a legal perspective is what what you're saying, and Dr. Yeadon is saying, though, it's very important in the United States, at least, to acknowledge the FDA approval happened. If you take a look at the BLA, the ingredients are redacted. If we acknowledge the FDA approval happened, and the left acknowledges that it happened, we're the ones who are trying to win this war and get the truth out, that are saying, no, Pfizer still has their EUA shield, because unfortunately, every crime is legal under the EUA. So willful misconduct is legal. So what you're doing wrong, it's in violation of state and federal laws, and the harm is greater than the good. Does fraud not meet that definition? Every crime is legal. So once we actually get the people to understand their rights and the law, then we can enforce it. What's happened is there's been so many lies about our rights and the law as well. And this demonic plan, there is a strange dynamic where they need us to agree with them with what they're doing. And the general population has been doing that. So if we don't acknowledge that the FDA approval happened, meaning the EUA shield was broken, meaning you have to tell us what's in those vials, that's the first step. The second step is to forget the VAERS database. Once FDA approval has happened, you can go into the BEST, the BEST database. You can subpoena the ICD-10 codes for every disease, injury, office visit, emergency visit, death that ever happened, with the CPT code of every vaccine for an American that was injected for 12 years old and over for Pfizer, and 18 of Moderna. And you will find billions of side effects. And that's the law. And if we know the law and understand the law, then we can move forward. So it's both a scientific, clinical, real-world evidence approach, and then it's also following the law as it is in the United States right now. And so we don't have a lot of time to acknowledge that this FDA approval happened over a year ago now to move forward, because they're passing new laws like getting rid of the animal studies, and then Biden has his executive order now to say that we need to be able to experiment or use human cells to produce, to change human cells the same way you program a computer. So that's the next phase. So if they get rid of the animal studies and we have an executive order saying, look, we can start doing experiments right into human bodies if we know we're going to do it and not do animal studies anyway, and believe we're under EUA law, they're moving forward, and we're not holding solid ground on past events that can easily be proven legally. That's what I'm saying too. And I'm acknowledging everything that the scientists are here showing and stuff. But there's, I think it's important to subpoena, or not subpoena, FOIA CDRH for the ingredients, and just go straight to Carl Mateus, the head of HHS, and get it from him. He oversees all of this.
Well, what we do know is that this stuff doesn't belong in there. What we also know that some of the stuff that was found, some of the heavy metals, of course, are extremely dangerous, are toxic. And legally, this means, just like Mike said, the injections have to be stopped immediately. But it also means that people need to be told that there's something wrong, because this is part of the informed consent. If you cannot consent to something that you don't know anything about, information is everything. That's why you're absolutely correct, Karen. FOIA may get us there, but I kind of doubt it, because as Mike and others have said before, the entire system is totally corrupted. They've had 30 years or more to infiltrate every level of society. And most of the agencies, most of the public agencies are not public agencies anymore. They're owned and operated, obviously, by the other side. So we're going to have to do our own research. We're going to have, as I keep saying, we have to do stuff outside of the system, because the system is totally broken. So what do you think, Shimon, this is about? You said that this looks like electronic circuitry.
Yes, the crystals that David has shown and I think Anna showed images of another colleague of ours, Matt Taylor. They do look like electronic circuitry. Mm-hmm. And I also have such images, but I also have other not less important images, so I would focus on those. It's not just electronics that we are seeing developing out of this. And the electronics, these findings, first, as Karen says, I can also point out at some very serious scientific papers, such as in Nature, one of the most prestigious journals. There is the technology to do that. And Karen has some patents, but patents are only a subset of the scientific literature. So there is definitely electronic circuitry in there. And in fact, you mentioned like Quinta Columna, they and others and myself, many people have replicated this strange phenomena that injected people start to emit something that is detectable in a very basic Bluetooth receiver, such as a smartphone, and it shows a MAC address that has no recognized device associated with as many jabbed people that you have around. This is amazing. And so this suggests that indeed there is something electronic in there, and not only that it interfaces and interacts with wireless electromagnetic radiation. And then both David and Matt Taylor have placed their samples in the vicinity of routers, and that they saw that electromagnetic radiation clearly has an effect on the self-assembly in the microscope. And then we see you just need to step into a room full of jabbed people, enable on your smartphone the functionality to show unrecognized Bluetooth devices, do a Bluetooth scan, and you will find a number of unrecognized MAC addresses as the number of jabbed people in the room. This has been replicated. No one's talking about this. So one thing I'd like to point out, in addition to what Karen said, had it been previous generations of vaccines, they would have been pulled out immediately after the first few deaths. Now, Reiner and Mike, I'm sure that you know that we already have, according to the different VERS system, millions of deaths of people worldwide and hundreds of millions of injuries, including severe injuries. And no, they are pushing ahead, full steam ahead. No one's even discussing withdrawing. Not only are they not withdrawing, they want to inject this into to babies. Now they want to combine this. I intended to use a profanity, to combine this with flu injections. There's no stopping them. So, and then I'd like to mention some more of our colleagues. We colleagues that are in several continents, we are doing our best to come up with the evidence that we can, given that we don't have any access to the very advanced labs that they have access to, and we only have mostly access to optical microscopy, sometimes to electron microscopy. We are presenting evidence that they, with all their labs and equipment and unlimited access to vials, are not bothering to show at all. So
I just want to say one thing, because what you're saying, because I think Reiner wants to talk about the legal strategy. If our team can just unite to say that these vials contain devices, obviously they contain devices, obviously they're electronic devices. They're not vaccines anymore. They can get kicked off the schedule. You were injected with a device, a medical device. It has different, that's my point. The point is that if we all acknowledge there are devices in here, everything that was protected under the vaccine law is gone. It can't get on the childhood schedule because it's a device. It's completely under different regulation. CBR had no authority to ever approve or authorize these. But the point is, people need to understand it's not a vaccine, it's a device that you're injected with. And that's where we need to get a unified message around that.
Well, we need to be able to prove this in a court of law. We need to have concrete evidence that shows this to a jury.
Well, PEG is a device. It's categorized as a device.
Yes, I understand that. We all of us know that this is really not, it can't be a vaccine, because a vaccine per se is something that immunizes you from the disease. And everyone, including the makers of these so-called vaccines, agree that it does not immunize you. So vaccines, they're not. If they are devices, as you're saying, and if we can show this in a court of law, then you're right. Then all of a sudden, we're on a completely different playing field.
Mm-hmm. Thank you. That's what I was saying. I'm a strategist, so I'm just kind of trying to get the big picture strategy, which is, can we all agree that these are devices, and that changes everything?
It could very well be.
I think
Shimon, is it the issue of these of the jabbed people sending out electronic signals that you can see via Bluetooth? This is a very controversial thing, because I know scientists here in this country, in Germany, who think this is not true. I know many people who claim that this is true, but is there any real evidence? Is there anything that we could show in a court of law that proves this beyond any reasonable doubt, so to speak?
Yes, I'm sure there is, Reiner. I can tell you that there are many people in the world, not just the ones that you know, that would love to discredit us. And they are, in fact, doing
it all the time.
They are doing it all the time. There are things that are very easy to demonstrate. One of them is the Bluetooth. You know, it's just so easy to detect. So I think that, we have those people try to discredit us and say that this is not serious. But the evidence clearly suggests that not only is it serious, but it's also very easy to detect and to demonstrate.
Mm-hmm. Well, you know, very recently there was an interview with a German doctor, I believe, who says that, and I thought this was a very wild story, who says that if you walk into a graveyard, and you, even there where you have people who got the shots and who were buried, you still can receive these signals. This sounds like Michael Jackson's Thriller movie.
It's true, Reiner. It's true. I saw there is a Spanish video about that. There are several.
Reiner, there was a doctor who did a study, and he questioned 137 patients, and 112 said that they were vaccinated, and 25 were not vaccinated. So then what he said, what he did was that he had them all turn off their cell phone. They turned off their electronic devices, and a MAC code remained on his cell phone that still detected an address. And so this occurred in 96 of the 112 people who were vaccinated.
Mm-hmm. And does he have any, it's not enough for someone to walk into a court of law and explain, this is what I saw. He has to be able to repeat this. Will this be possible?
Reiner, this is easily repeatable and replicable. And I think quite a few teams, you can do it yourself, Reiner. This is very simple.
I'm not going to get the shots.
No, don't get you just need a cell phone, enable the scan from Bluetooth, unrecognized Bluetooth devices, and you'll see the results yourself. It's very easy to do. I would just like to add to that those usually fortunate people who do give off the MAC addresses are those who are alive. Most of those who are in the grave, unfortunately, don't give off the Bluetooth device because maybe they perished before the circuitry developed in their bodies. So some of the things that are in these technology do not generate electronic circuitry. They generate other features that I could expand on that are toxic and deadly. And the electronic circuitry is rather the more benign, albeit horrific part of what we are seeing.
We're going to need someone who does the equivalent of the autopsies that Professor Burkhardt and 50 others, I think, have performed in order to show and have irrefutable proof that this is really the case, that this is really about electronic signals or other signals being sent out by those who got the shots. What about the interaction with microwaves or 5G that you've been talking about, Shimon?
So the electronic circuitry interacts, so we see, with electromagnetic radiation in two ways. One way, the first way, is that the electromagnetism, as well as temperature, ambient temperature, and other factors that Mike mentioned earlier, are affecting the dynamics of this self-assembly procedure. And I think David showed remarkable things, like that they can even delete a circuit once it's being assembled. So, and then recreate it. So this is the first interaction. The second interaction is once this circuitry is finished and ready, it can communicate with and generate these Bluetooth devices. And this is very worrying. So functionality of self-assembly and then functionality of actual communication. But I want to point out that the fact that I have images of additional features that are not electronic circuitry, also affected by electromagnetic radiation in their self-assembly, and these features could be ribbons, filaments, tubes, very interesting features indeed.
Can you share those?
Yeah, I can share. I mean, I prepared two presentations. So this is going to be the second one, but nevertheless, it's at this point it's adequate to show. So let me share the screen, and I'll show you the second presentation that I prepared. Okay, so these are also dark field microscopy images, very low magnification, because these features, these ribbons and structures, the crystals, they're very big. They're almost, actually, if you look at the glass slide, you can see them with the naked eye. So there might be, for example, 100 microns or 0.1 millimeter in size. The interesting things about these structures is that our colleagues and Anna here also, we have their counterparts that have been extracted from the blood of jabbed, and also from the blood of unjabbed people. But they correspond very well what we see happening. So this is what we would call a ribbon. It's a very complex ribbon, and it's self-assembled. There was a drop of liquid from the vial, and then I placed it near a router, and after 24 hours, all these things self-assembled, and I'm going to show you.
So this is not nanotechnology. This is more like microtechnology, right? Because it's so big, the structures.
That's the names are confusing. The nanotechnology is what self-assembles these structures, but by the time that they are finished, this nanotechnology could produce millimeter-size features. Mm-hmm. So if you were to look at this with an electron microscope, you would probably see a lot more detail showing how this is self-assembled. But yeah, nanotechnology can and does, in fact, produce almost millimeters, but definitely tens and hundreds of microns in size structures. Let me.
But what is this? Is this something that shouldn't be in a vial, and of course not in the human body? Does this not happen in nature?
It's not in the vial, Reiner.
Mm-hmm.
It's on the glass slide that the microscope is looking at. So what you're seeing has self-assembled after a week or so on the glass slide that I put near a router, again, to add some electromagnetic energy into this process. And then, if this self-assembles after a week from a drop of the injection, you could, I think, it's rather straightforward to assume that these things, I'm trying to move forward to show more images, but there are some captions here that are I don't know how to get rid of these. So let me try this. Yeah. Okay, here is another thing. This looks like a tube. If the previous one was the ribbon, again, you can see that not only did this self-assemble, but it left margins that are blank here on the glass slide. And these are probably it took the material for its self-assembly form. So you can see that all around is black, and then there is a lot of debris around. And this is a recurring feature that I will show. Well, by observation on these features that look like ribbons, tubes, filaments, they seem to be carbon-based. I would call them artificial organisms, and some of them could be filled with some toxins. Now, the reason why we think that these are carbon-based features is very simple. Whenever we do a electron microscopy on this, the two most obvious elements that we find are carbon and oxygen. Sometimes we find a little bit silicon, a little bit sulfur, and maybe some of the metals, but these are mostly made of carbon and oxygen. So hence the assumption of graphene oxide, which is carbon and oxygen. Now, the reason why we don't see the graphene oxide when we look at it inside the vials is because it's being sequestered by the lipid particles of the hydrogel that I can show you later in another presentation, holds and sequesters everything inside. But once self-assembly occurs, all these things open up and release their building blocks to allow these structures to be self-assembled. And Reiner, even if it doesn't carry any toxin, imagine 1 millimeter or 0.5 millimeter structures being assembled in your veins and arteries or in your brain. This is not good, is it? Of course. And these same structures are being pulled out during blood analysis, mostly of jab people, but now, unfortunately, we are seeing the same ones in the blood of unjabbed people, such as myself, I think maybe Anna, and others. So this is why we are talking about the shedding, not of any imaginary virus, but the shedding of this technology. And we have other colleagues. We have one colleague from Asia who is researching. She's actually culturing, believe it or not, these. So she can filter them out of the liquid, and you place them, and in capsules, and they grow. They continue to grow. Somehow, it's as if they are like sucking carbon from the carbon dioxide in the air, this is really scary, but unfortunately it is very real. Let me show you a couple of more. Okay, this is a larger magnification, so you can see that this is not very smooth. It has innate structural features in it. This is a particularly scary one because to me it looks like an artificial organism that has spouts, and you can see the spouts, the nozzles here and here and here and here. It's as if it was designed to discharge, and maybe very slowly. So I'm speculating a little bit here, the contents that flows inside of it. And again, you can see that it's self-assembled and left a blank margin where it took, I think it took the ingredients for its self-assembly from, and this is the blank region around it. Again, this is a tube. It has a mouth, an opening. I can show you other tubes. This is classic. This is what we call a ribbon. Again, when we inspect this with electron microscope that is equipped with EDS, a spectrometer, we see mostly carbon and oxygen. So it could be graphene oxide, it could be graphite. You can call it carbon. I don't care. And the interesting thing about this is such structures are exactly what we see in people's blood. Just a few more, and another tube or a worm or whatever, artificial. This is a tube. You can clearly see the opening. Here it is magnified a little bit. Amazing, Reiner. So the variety of features, this to me looks like an artificial organism. Again, this is nothing biological.
Well, here's the thing. I know because I've seen the video, and I know some of the people who appeared in this video, that German doctors who examined the blood of those who got the shots found similar, whatever it is, similar things. Two of my colleagues were with them. One of them, who I closely work with, is an attorney, and they all agree that this stuff doesn't belong in there. We all agree on this.
It doesn't belong in the. It's not in the vials,
and it's not a natural thing that could occur in people's blood naturally.
No,
that much we do know, I think.
And I also have images of electronic circuitry, but the ones that David has here are better than mine. So there is a plethora of structures that self-assemble. And again, let me just emphasize again, this is not in the vial. If you look at the vial, you don't see these things. You have to have a lot of patience, put it on a glass slide, an incubator, or maybe just let it sit for a few days. I actually tested this thing with an actual incubator of body temperature, and it did speed up the self-assembly by quite a bit. And this is quite scary, I would say.
Yes. But it is in the vials. It's just that you can't see it at first. It needs to self-assemble first, right? Because if it wasn't in the vials, it wouldn't be in people's blood.
This is not in the vials. The technology that results in this is in the vials.
Yes, right? Of course.
Shimon, can you show the images of the self-assembly from the liposomes? Just because we're talking a lot about the liposomes, and you're the one who's captured what actually happens to them. So I think that would be a very important point.
Okay, yes, I can show that. So speaking a little bit about the chain of custody of how we got these vials. So in most cases, we are using used and open vials. Sometimes vials that have been standing in room temperature or exposed to heat, not according to the specifications of transport of these things. But in one particular case, we managed to get in Europe sealed and frozen vials. So we will not disclose how we got them, but the chain of custody there was such that it was clear that they were transported according to spec, frozen. They were sealed, so they have not been haven't been opened yet. So, Reiner and Mike, these are examples of sealed Pfizer and Moderna vials just upon thawing. So this is a unique opportunity because I think nowhere else has anyone managed to just look at freshly thawed vials to see what comes out of them. And this is looking at them in just ordinary bright field microscopy. So the contrast is not as impressive as in David's case. I'm going to show some phase contrast images later. But what is obvious is that you can see very large, maybe there are liposomes, right? Here is, I can zoom in on these. This is something that we should not have seen at all using an optical microscopy, because had their narrative been accurate, namely that there are lipid nanoparticles in these vials, nanoparticles imply that they could not be resolved with an optical microscope. So what the hell is a 10 or 20 or 50 micron, and not one, there are many liposomes doing here. They shouldn't be here at all. Again, this is reason enough to stop this immediately, but I can show you some scarier things. One interesting thing is you can already see in this particular case at room temperature, this is not using any incubator, you can see something sprouting out of one of these spheres already. Okay, so, look what happens. This is a Moderna sample. This is amazing. When we waited, when we had the patients to wait, we see that some of these large liposomes popped and burst. And what was inside them was amazing. We found that they have nested liposome inside them. Here are some of the spheres, and you can see the needle-like shapes, but clearly more spheres in another case. And also this yellowish stuff that spilled out from them. So these are containers, these are capsules. These are like matryoshka dolls, Reiner. There is nested features, and this matter here cannot be resolved. Maybe it's a hydrogel that I wanted to discuss further, but we can just see that there is yellowish matter that, again, shouldn't be here. What is it doing here? This is not mRNA. And interestingly, when we let these features dry out, like in this image C, we can see the needles and the hydrogel or whatever matter it is, but we can no longer see the nested spheres or nested capsules. They also popped and discharged yet more material into the medium. However, some of the spheres or the liposomes were so stable, unlike those I just showed, that they remained intact even after a week on the glass, and they did not pop, did not burst, and this is what you see here. Moving forward, this is just an image of something we use to measure the sizes of this. This is moving to phase contrast, and this is from a Moderna sample, and the two images A and B are just normal bright field microscopy, not impressive contrast. But when we put the freshly thawed liquid inside this chamber, this is 50 microns. So you can see that there are features here that are 25 microns in diameter. and they shouldn't be here at all. We shouldn't be able to see such large features. But the real thing happened when we, in this case, not dark field, but rather phase contrast, which is yet another contrast enhancement technique used in microscopy. This shows, this is the bluish, the colors are not real. These are colors due to the phase changes in the image. This is not a video like in David's case. This is just time-lapse images at 5-minute intervals. So after 5 minutes under the microscope, these lines are the reticle of the counting chamber. The liposomes are more or less intact, although you can see that there are different kinds of them. This black one is not the same as this yellow one, I would say. Now, this is what happens in 5-minute intervals. This is unbelievable. So you can see the self-assembly starting to happen at room temperature, and this is 5 minutes, Reiner. Not more, not a week, not a day, not a month. 5 minutes at room temperature, and I think that what we're seeing here, we can identify the motifs. One motif is the you can see the very fine filaments, and then there is the these are 2 beads. I can show you more beads here. Here is 3 beads. So and this is another filament, more obvious and larger. And finally, we can see chains of beads assembling and ribbons, maybe this and this, and then fine, very fine filaments. Here is a more impressive ribbon, and you can see that they are coming off the spheres at their end. So these containers, the capsules, are providing both the hardware and the software, so to speak, for the process, the blueprint, and the building blocks for this self-assembly. But it gets even more interesting when we go to Pfizer. A similar freshly thawed vial placed in the counting chamber, and you can see here these species of particles that are very large. This is one that I will show more later. More of the Matryoshka thing that I discussed earlier. So things are bubbling up inside of it and are starting to discharge. This is very scary, Reiner. I don't know what soup is cooking, what is brewing inside of this. This technology has a name. It's called multi-compartmental polymer zones. This shouldn't be in a nanoparticle RNA allegedly injection. And again, when we look at with phase contrast at the self-assembly, you can see that in the case of this Pfizer vial, it happens much quicker. After 5 minutes, we already have this crystal. And again, a plethora of filaments, such as this S-shaped and this one. And the beads that I mentioned earlier here and everywhere. And this is growing. It took it 5 minutes to start growing. And then if we wait 5 minutes more, it becomes more pronounced. Sorry, this is, I made a mistake. This is 10 minutes. No, I was right. This is 5 minutes. This is 10 minutes. And you can see the crystal here. And the most interesting one is this one. And you can see the different motifs. So this particle here has sprouted to both sides, and this is like a linear filament. This capsule has sprouted to a chain of beads, and this one has sprouted to something that is curved and bent. And then we have this thing here, and I think I have an image dedicated exclusively for it, because what we see here is again something that only phase contrast. No, not only. In fact, if we go back to revisit some of David's videos, we can see murky clouds present, and this could be lipid nanoparticles. But in this case, you can see that these are not just lipid nanoparticles. Obviously, they cannot be resolved by an optical microscope, except if there is a big cloud of them. So this is the interpretation that I can give to what we are seeing here, that looks like a foreign cloud floating in an otherwise rather uniform medium filled with filaments and beads. Okay, so if we later go back, we can see this murky cloud in the dark field microscopy of David. So this is corroboration that there is something that is gel-like inside these vials, and here is a phase contrast image of it. Going forward, this is just image enhancement of images that I showed you earlier. I just wanted to show that there are different type of large liposomes. For example, this one is transparent because you can see the reticle line underneath it, whereas this one, for example, this yellowish one is not transparent because it obscures the reticle line. And then this is a multi-compartmental liposome because you see there are features inside of it. And this
what
is
are the lines, Shimon? What are the lines we're seeing?
The lines are simply the reticle lines of the counting chamber that I showed earlier.
Okay.
This is the reticle. That this is a pool. The pool serves a few purposes. First, it provides more material, so this doesn't dry out very quickly. And then at the bottom, there is this reticle that you can see here. And this is for measure, and you can see what sizes these lines are. So it's a scale, basically, to give us an idea of the size of this. And this is the Pfizer one that I just enhanced the bubbly soup that's brewing things that coming out of here. I think maybe Karen Kingston also could later show. I can also show not patents, but scientific papers about this technology. This is a technology. It's not just a coincidence that it's in
That's what I was going to ask. What are we getting at right now? We've seen the images, we get the idea, but what's the technology behind this? What does this stuff do? Why was it designed?
I'll just real quick. I haven't looked at it in a while, but I can pull it from last year. This patent talks about using SRF, which is basically a vascular endothelial growth factor substance. So it's for embryonic development, from what I recall. But I'll send it to Shimon after.
Okay, so my research found that the science of lipid particles, I wouldn't call them nano. This is Reiner. You can see this is 50 microns. This square. So this one is the largest we have found in the Pfizer vial. It's 50 microns in size.
It's pretty big, yeah.
Pretty big. And this is called, in the literature, it's called multi-compartmental polymersome, or they could just call it liposome. So basically, it's a matryoshka configuration where one capsule can hold, I referred to it earlier, can hold materials, and it can hold them in different compartments. It could even mix them just before discharging them, and while keeping them separate internally. Reiner, the options here are unlimited. They have the most amazing technology here. And let me just move on to show what other things we found. So this is just an enhancement of this image that I showed earlier. Sorry, this one over here. It's just basically contrast enhancement. So you can see people are discussing what are the materials in these vials. What I wanted to show is that the large spheres or liposomes. There are different species. You can see this. This is not dark field. So what you see here is bright field. This is the exact color as it looks under the microscope. So you can see all different colors and sizes. There are brown, brownish, blackish, yellowish, whitish. But
other than that, this stuff certainly doesn't belong in the vials. We don't know what it's doing. I mean, it's easy to imagine then when people get injected with these things, or the stuff that produces what we're seeing, it's easy to imagine that this causes blood clotting, for example. But other than that, I have no idea. Does anyone have an idea?
So if you have these structures developing inside of people, this is not good. We could discuss later, and we could maybe have some of our colleagues come over for another meeting to discuss. These structures in themselves could be thrombogenic, and they could contain inside of this filament could contain inside of it other things, and it could contribute to shedding. But this goes too much forward. Let me just show you. This is the incubation we did at 37 degrees, which is body temperature. And we had, in this case, 4 samples of BioNTech or Pfizer and 1 Moderna. And we compared it to control, which is PBS is just phosphate buffered saline. We expect to see nothing in here. And after 4 days, we could see very large structures. Again, 0.2 millimeters is this size, and this is much larger here and here. And you can see, although the resolution is not high, that in addition to structures that are assembling and are very large, Reiner, this is like 0.2 millimeters, there are also the spheres that seem to be awaiting yet more energy or more activation signal to develop further. And in the case of Moderna, it's very interesting. We see the exact same structures, but in a smaller scale. So if I magnify this, you can see that it sort of resembles this. And I will show you again, this is the same thing after 7 days of incubation. Again, this is phase contrast. So you should ignore the colors, just view the contrast. And this shows the variability between batches, because we see that in the BioNTech Pfizer, so number 1 and number 2, number 3, they have amazing structures developing here. None of these structures should be in the blood or tissue of anybody. And the number 4 has, the motifs again are needles and crystals, but we also have things that look like electronic circuitry. So if I go
as I said before, what is astonishing to me is that what you found in the vials is exactly, or pretty much exactly, the same as what was found in the blood of vaccinated people by, among others, this colleague of mine who I really trust. But we still don't know what it does, except, of course, it's easy to imagine that it's causing thrombosis.
May I, Shimon, may I just quickly comment, and Reiner? I think
Sure.
what you've just shown me here, I think, is in and of itself sufficient as part of a sort of 3 or 4 paragraph strategy, which Karen would be much better than me to lay out. But I'm happy to be, whatever, helpful if I can be, while the team tries to work out what it might mean. And I know each of you have laid out what you think, but I think this alone, I would say, is it's good enough for prime time. You could describe it to a man, woman, child in the street and say, this is concerning. These materials, these shapes should not be in this medical product. And given all this other stuff like the VAERS, and then maybe even my backdrop to why vaccination is not an appropriate strategy, I think that's a non-conspiratorial thing to lay on people. Whereas I think if we go to self-assembling electronics and so on, might be true. And if anyone would suss it out, people on this call and a few others would probably work it out. I can feel I would have an immensely difficult job, even to persuade people who I regard as on our side, because they would just raise all sorts of objections. So I'm wondering whether, if we were to calibrate where we are, we could say we demand these are withdrawn, but pending that, the FOIA stuff on ingredients is important. And I think Karen, was talking about the classification of these medical interventions are inappropriate, reference, and so on. And now multiple people have identified, both from the vials and occasionally from the blood, concerning structures. And that seems to me, we'll put it this way, it's the smallest tinfoil hat you could possibly wear, because it's all true. Does that help at all? I mean, it seems to me that...
Yes, I think that's very helpful, because if you go beyond this, because I've noticed that if we go beyond this, we will not get very far. We can only speculate, unless, of course, others will join and help us. That's why it's so important to spread this information to as many people as possible, because I'm sure there are other people out there who may be helpul in this. But
one other thing, if I may, before I forget, Reiner, Shimon, I too have watched a couple of video presentations about MAC addresses from unidentified devices, and it's really scary. I read the entire canon of wonderful post-war US sci-fi in my teenage years, so this is not a particularly, it's not even that wacky a plot if the technology would allow it, okay? So I don't think it's that strange, and I'm not reluctant at all to toy with these ideas. But I think if we go beyond that rather guarded set of statements, which I think are powerful enough in their own right, if we go too far, what happens is I have found people, I just lose them. Lose them. They're not interested in nor able to look at what's being shown and said objectively. And as much as we would like them to, my own experience is I have relatives, for example, with PhDs in biology, and I've been unable to persuade them that anything is wrong at all with the narrative. And they know their brother has had multiple decades of biopharma experience, did quite well, but have persuaded themselves, and I show them and I cannot get them to move. Now imagine how much easier my job would be if I said, oh, by the way, it's self-assembling electronics. But the door I would even feel the whisk of the door going across my nose. So I think, so I'm on your side. I'm on the side of I know the people here on the call and elsewhere are absolutely sincere, very as troubled as we are all equally troubled. And I guess it's a question of the legal strategy, the FOIA strategy. Again, I'll shut up in a minute. By the way, there's a small company called Judicial Watch or institution, and I don't know whether you know them, but Bill Marshall has been just very good at getting information out of the right authorities. So maybe we could play to our strengths and work with Bill and FOIA something, because again, Anna and go on. I'll shut up because I could go on.
So I have, I don't know if you want me to show on. I have a paper. I have several papers that address what you're seeing. I can share real now, or I could just send them to you. I have the FDA database that shows pegylated lipids or devices. I can maybe do a quick preview of that. It's not, it's in their databases. It's in the filings with Pfizer. Do you want me to show you that?
I believe you. I absolutely believe you.
So let me show. So I can show. Do you want to look at it real quick?
Let's take a quick look, Karen.
Karen, before you begin, let me just please briefly address what Mike has just said. So, Mike, I think you're right. We could choose what level of depth to go into when we present this to people. When we present this to a more educated scientific community, we may choose to go deeper. And when we present this to laymen, we could choose to limit ourselves.
Yeah,
but I think, Mike, there is the notion. I simply have to share with you my impression that in the last 2 or 3 years, something has happened to the cognition of people, including mine. I feel that I have cognitive impairment. What can I do? But some people seem to have undergone some kind of lobotomy. One of these participants here used this word. They cannot understand or comprehend what is told to them.
Yes. I'm not sure whether it's won't do it or can't do it. So, but I agree with you. Something we've ended up with like a brain block. I know some people, just literally 30 seconds, who know my background, who believe that I was a very normal person, would watch the national news and read the broadsheet papers and so on. So they knew I was just an ordinary person. I worked at Pfizer. I began to describe what was going on. It's maybe 3 or 4 months into summer of 2020. And within 5 minutes, this woman's face had gone completely white. And she's staring at me. And I said, what's wrong? And wordlessly, soundlessly, she said, I could see her face, I'm scared. I think she said, I'm scared, soundlessly. And then I broke the spell, and she said, if the truth anything like what you're telling me, it's so frightening, I don't want to know anymore.
Mm-hmm.
And I think that I've got that from lots of people. So I think even if we could defend a little bit further than what I summarized, I just don't think it would be productive. People will flee like, oil and water.
I agree.
But I think what you've got.
The strategy I'm talking about it's not scary. I mean, it's disturbing, but it's not scary. It's just that pegylated lipids are devices. They've been classified.
Help me understand why that isn't just, they've looked at, some of the components are devices, but altogether we're going to call it a vaccine and take it down this other hole. You may be right, they did the wrong thing, but in the end, so what?
People don't know they're injected with devices. I'm saying it's a very clean strategy for recall and injury. You weren't told you were injected with a device.
Okay. Yeah. No. I just wanted to know why it mattered.
Well, because it's not terrifying, and it's something that wasn't disclosed to people, and it's absolutely not a vaccine because devices are CDR8, as you know. CBR is vaccine, so it's just a very clear.
Yeah, it's a complete mess. So I understand.
It's a complete mess. It's a very clean.
Yeah. I know. We're on the same page.
And I have other stuff I'd like to share with the researchers, like the studies to validate what you're seeing under the microscope. And I'm happy to email them afterwards. But I'm saying this strategy is just a very clean. Everyone knows pegylated 2000s in the Pfizer vaccine. That's a device. I can show it to you, so we don't have to. It's very simple.
Is it necessarily true that a complex product containing, I don't know how many, a dozen ingredients, if one of them is a device, is the product automatically a device?
No. So I worked on Theracos photophoresis, which uses electrophoresis and methoxysorolin, and the device is classified and filed separate from the methoxysorolin.
Yeah. So it's already I understand what you're saying.
The device has to be filed separately from the ingredient. I know this from my personal professional experience. Yeah.
Sure. No, I absolutely believe you. Yeah, that's fine. It's just what I imagine they'll probably do is to say, there are 10 components, one of which in isolation does classify as a device, but we judge the overall product to be whatever. And, then it becomes a legal matter. It's not even very powerful. If I tried to explain it to someone who was not technical, had no regulatory experience, they would look at me and say, well, as long as it's been assessed correctly, Mike, I don't really mind. That's what they would say. So
the pegylated lipid would be one of a couple other things that are not classified as biologics by CVR. And then you would FOIA the rest of the redacted ingredients.
Yeah.
I think it's
a good one. It's a good one. And again, have you yourself had the resources to pursue some FOIAs?
No, I haven't found an attorney that's willing to partner with me on it.
Sure. This Bill Marshall guy, Bill Marshall of Judicial Watch, Tom, I don't know. I've spoken a lot to Bill, and Bill also knows Sasha. So he's keen as mustard, and I think if we had some, we had a simple strategy that was simple enough that we can explain it to people, and that's what it would need to be.
I can show you now in like 5 slides if I can share my screen. Okay.
Yeah, I absolutely believe what you've just said. It's going to be the question of what are we going to be able to do about that, given the corrupt institutions.
Can you guys see my can you see my screen?
Yes.
Yeah, a little small, but yeah, I can.
So anyway, this is the FDA approval for Comirnaty. There's the lipids there. You can see right here, there's polyethylene glycol. You see it, 60,000? Yeah. This is also in the BLA, the polyethylene glycol 2000. If you go to the database for devices for the FDA, you can see, I'm sorry, I can't see my full screen right now. You can see that these are for devices. Polyethylene glycol is listed as a device, 2000. This one is called Alpha Hang PEG. Again, it's a device. As you can see here, the pre-market approval is not released under Section 814.9 21 CFR, the FDA medical devices. If a product is used in experimentation and has not been FDA approved yet, it does not need to be disclosed to the public until the FDA approval is granted. So this is protected under intellectual property. So here's another one, another PEG 2000. This one, I believe, is the American Red Cross. As you go through here, you can see also it's not pre-market approval. So they're not telling you all the ingredients, what it's being used for, because it's currently under experimental use and protected. But if you actually go to the website for the Red Cross and go into their CAS filings, you can find that it's listed there. It's harmful. This 29 CFR 1910 is an OSHA warning, meaning that you're not supposed to have skin contact with polyethylene glycol 2000. That's how it's categorized. Clearly this should not have been put in there. It is categorized as a device.
And I guess those components, because they are not GRAS, they would have to be separately qualified through an appropriate toxicology program, which they have not been. So straight away, that's like 8 levels of concern. Also, as you know, we know that some PEGs, depending on molecular size, are commonly associated with allergic responses, anaphylaxis.
The point is that it's a device. And as I go through the ingredients and I go through the databases with the FDA, these are categorized as devices or toxins not for human use. So I'm saying it's in the ingredients listed in Pfizer. It's a device. It's not. I mean, what they're calling lipids are devices and toxins. They just because they called it a lipid doesn't. I mean, does anyone understand? I mean, I feel like
it seems clear to me. Just because they call it a vaccine, it's not a vaccine. It's
a device.
Yeah.
And all this other stuff I can validate too, but this is just a very simple. It's not only is it like not only is it not a vaccine. The regulatory body that oversaw all of this never had the authority to do so. It's a completely different regulatory body under the FDA.
Yeah, this is definitely in and of itself extremely scary. And as Mike said, we can go beyond this, but we need help with that. Our task should be to get this information out and to get it and to spread it to as many people as possible, because there are others out there who will be able to step in and help. And there will be attorneys who will help you on that FOIA stuff too. This gets scarier by the minute, so to speak. And if you just look at the device or at the structures that Shimon just showed us, and what I've seen these very same or almost the same structures in the blood of people that have been examined by doctors who I know and who I trust. If you look at this, it looks like technical devices. It doesn't, and it could even be, but this is again moving into speculative territory. That could be electronic devices. But definitely, I agree. These are devices, even by the look of it. They're devices, yes. But what do they do?
And Reiner, I would like to share just from a clinical physician standpoint of what I'm seeing and what I can show you in live blood analysis in terms of what Shimon has shown. Would it be okay to do that? Or Karen, do you have other stuff to share? Because I really want to hear what you have to say. If you do.
No, I have additional information to support what Shimon just showed, and I'll email you those documents. I have not had a chance to go through and review them, so I don't want to speak about something that I haven't had a chance to thoroughly review. But I'll send you the publications.
When you email this to us, we're going to put it up on our website, but when you email this to us, before you do that, can you try and decide what you think this means? Because if we're left to our own devices, I don't think I'm
going to email Shimon the
okay, good.
I can show you what I'm going to send a publication and he can go through it. That's entitled Stimuli Responsive Nanoparticles, Nanogels, Capsules for Integrated.
Okay.
Let me just share my screen real quick so you can see it. I think, and then this is what I was going to send. Hold on.
This would be a good opportunity to thank you, Reiner, to bringing us all with Karen and Mike Yeadon under this show of yours, and we can get to network with each other. And it's nice to make your acquaintance, Karen. I really appreciate your work. Thank you.
Yeah.
Thank you, Shimon. This is what this is all about. We need to connect with the people who we can work with, because this is what I keep saying. And when you alluded to the fact that maybe some people have lost their minds, this is my terminology, of course, I'll remind you of what Professor Susharit Bhakti and his wife found out, how this seems to be affecting the minds of the people. And when you use the word lobotomy, that is exactly what Dr. Peter Breggin said. It reminded him, he's the one who abolished lobotomy, which is one of the worst crimes against humanity ever committed. Most people don't know what this is, but if you have seen the movie One Flew Over the Cuckoo's Nest, then you do know what I mean. So Peter Breggin says it is as though it doesn't work the same way with everyone, but in some people, it looks to him as though they were lobotomized. And Susharit says it breaks the will of some people. And that's why I think we shouldn't be concerned about people who don't want to talk to us or can't talk to us. We should focus on our own group, on the group of people who are willing to find out what's going on, and who are willing to do something about it. All the others, they can join us if they feel like it, or if they are beginning to ask questions, but we shouldn't focus on them. We should focus on our own group of experts, because they're the ones, we are the only ones. We're the cavalry, basically.
Yep. So just to validate what Shimon was showing and what all the scientists have said, this is what you're looking at. It's not one technology, it talks about nanoparticles, nanogels, capsules. Also, the pegylated lipids are hybrid with, some of them are proteins that are hybrid with quantum dot. I have that study as well. But this one talks about, this is just a general, what you're seeing is hybrid nanostructured particles from nanometers to a few micrometers, microgels, core shells. So this is core shell structures. And then I have a book chapter on genetically engineered magnetic hydrogels that talks about how they take nanotechnology to build out micro and macro size structures. So this is probably what, Shimon, you're looking at, is this technology. It's some of what it is. So it is intelligent. So it is self-assembling, but it also responds to frequencies and signals. It responds to its environment. So I can share this information with you.
It has this nested structure, and I can definitely see this in the slide that I was showing. Thank you.
Yeah, and then an important point that I didn't share before, I wanted to share real quick about the pegylated lipid is what it is used for. Yeah, here it is. So this, we were told that this doesn't do gene editing. So this is the Broad Farm is a supplier to Thermo Fisher and other companies. And it says here the PEG 2000. Okay, So this is the PEG 2000. So this has already been disclosed to us. This is my point, is that I have what's already been disclosed and filed with the FDA. The PEG 2000 is used for liposome preparation, right? And again, we're told this wasn't doing gene editing. It's for small interfering RNA. It's for silencing genes inside of our body. It says it right there. And it's delivered in the COVID-19 vaccines. So siRNA is used for gene editing. That's what it's for. And this has typically been used in anti-cancer drug delivery systems, So this is normally...
I absolutely get it.
Okay.
I think, Karen, in a way I didn't before. Quick question. Do you think if the product contains, say, 50%, whatever it is, DMG PEG, it's a device, I think we're going to say, what happens if it's like 1%? Is there a point at which
I believe it does? It's more than that. But Mike, I have other products that are disclosed that are also toxins and are supposed to be used for in vitro gene editing. So this is not. So I'm only sharing you.
Gotcha.
I have done thousands of hours of research. That's probably equivalent to tens of thousands. And I'm not. Yeah. So I need to be treated as a partner, though, not as an assistant, which is kind of what's been happening. I need a strategic team that's willing to partner with me to get this information out and not say, can you find out about the luciferase? Can you find out about this? That's kind of how
absolutely. I think my first question is, it seems to me it doesn't matter. Well, it does, but it kind of doesn't. If I'm a person in the street, even a physician, say, who's using the vaccines, I probably don't really care if it's been approved by federal sub-branch A, B, or C. And now I know you do.
It matters from a legal
Why does it matter?
It matters from it matters from a legal perspective. One is because so if you're going to argue in a court of law that this can be on the vaccine schedule for children, no, it cannot. It's a device.
Mm-hmm.
It's a device.
Okay, so are we saying devices can't be vaccines? I'm just acting
difficult.
difficult.
No, they can't be vaccines. Vaccines are biologics.
Mm-hmm. This is a new vaccine, Karen. This is new. So, I just don't know. You're absolutely right, but I just don't know if the person hearing the case was corrupt, they would just be able to back you out of court by saying, well, this is both a device and a vaccine, and we've decided to classify it as a vaccine. Got to go somewhere.
Yeah, but I think, I'm hoping if enough people haven't had the lobotomy yet, that they'd be offended that they were injected with a device.
That's the point. We have to get this out. We have to get this information out. If people see these pictures, I've seen them before, and I thought, well, why isn't there an outcry? Because I was assuming that everyone, including the mainstream media, had access to them, but it's not true. It's only us who have access to these pictures. I think these pictures are in and of themselves so concerning that anyone who hasn't been lobotomized yet, who has half a brain or a pulse, and who has gotten the injections must get extremely concerned about this.
Yeah, there is no, I often use this phrase, there's no benign interpretation that can be placed upon the observations. Seriously. So, you don't need to be a lawyer or a philosopher. It's just, there's no benign interpretation of what they've done and are continuing to do in numerous dimensions. And that this is an important one.
Your point, Karen, is, if I get you correctly, if it's a device, it cannot be a vaccine. If it's a device, whatever authority has allowed this product to go on the market was probably the wrong authority, because there's another authority out there which is in charge of devices. Yeah. Is this correct?
Yeah, and the first step too is, like, if we just jump to, you were injected with self-assembly lipid nanotechnology that's building out, viruses and new creatures inside your body. Whoa, that's a lot. But if we can start with, this is a device, and if people can acknowledge it's a device, and then we can show, and here's what the device looks like inside your body.
I keep saying that it's not the courts of law that will turn the tide here. They're going to clean things up with our help. We're going to need the courts of law, or our own version of the courts of law, if we can't trust the ones within the system, to clean things up and to take these global corporations that are responsible, to take them down. But it is the people who we need to convince. That's why it's so important to get this information out. And but Mike is correct, because if you are going to take this case into a court of law and you're inside the system and you have someone who is corrupted to a degree or under pressure, then this is probably going to be their line of defense. They're going to say, yeah, well, maybe it is a device, or maybe part of it is a device, but this is a novel product.
Yeah, but I'm saying that at least you get the acknowledgement that it's a device.
Yeah, I understand.
And then you can go and say, this is what the device does inside your body. Now, I only showed you one of the devices that's in there. 80% of the devices are redacted.
Mm-hmm.
I have other FDA filings on QDOT that currently are not public available. Is QDOT in this technology? No one can say whether or not it is, because the other devices are redacted. But we know this one device is in there, and then we're observing these additional devices. I'm trying to come up with a logical, factual, what people can digest, right?
Yeah, and we need something that people can understand, because we need to talk to the people. First to the people, and then to the courts of law.
First to the people, and then the courts of law. But let's first say this is the device. This is one device that we know of. And here's these images of all these other devices. We can go to the BLA. Look, they've all been redacted. We don't know what these are. We have some evidence, and we have strong hypotheses on them, but we need that information from the FDA, from CDRH, or whoever it's been filed with, and the manufacturers.
Yeah.
Yeah. Does that make sense? What I'm trying to do a
You're trying to put together a strategy. Yes.
I'm trying to put together a strategy. Yes. Yes. Does anyone have comments?
Yeah.
I really think that the strategy is multi-level, and I think that Karen's strategy is extremely important, and the researchers are extremely important. I think that what I've always loved about when Karen Kingston speaks out is that she puts it in the broader context of what we're fighting for is the human species, and getting back to the point that 5.3 billion people have been injected. We don't know how this technology, for example, is related to the phenomenon of died suddenly. Are the long clots that are 6 feet long, is that part of self-assembly nanogel or carbon tubes or anything like that? And then the next thing is that the phenomenon of shedding that is still being denied, even by frontline doctors, is for real. And I would like the opportunity just to show a couple slides just to really show that this is an issue, because if we do not acknowledge that this is a societal problem and that we literally have people with long COVID who are uninjected, who have ongoing problems, and the regular doctors are saying, oh, this is because the spike protein is circulating, etc., but we're finding the same structures in the unvaccinated blood. And it's correlated with blood markers, for example, the D-dimer, which shows microclotting on a massive scale in unvaccinated people, women who have hemorrhaging, we have people with headaches and blood pressure abnormalities, all of these things. And if you would just allow me, I just want to show a couple slides just to show you what I am speaking of. And just one second here. Are you able to see? So I got the same microscope as David, and I've been working with him via Zoom on his studies. But I really work on also finding out what is the impact on the unvaccinated in the shedding phenomenon. And we don't know whether or not these structures are also environmentally coming to us. For example, through chemtrails, through the food supply. Clearly we have these 26 teams worldwide who have found these abnormal findings. The RNA sequences we discussed, that there's fragments. Some vials have no phosphorus or nitrogen. We have DNA and protein impurities. If you just have broken RNA, that in itself causes a huge problem. Then we discussed that these huge amount of metals are there. Cesium is radioactive. So do people have radiation poisoning from that? large structures are being found and forms of graphenes are possible. So this is an unvaccinated person who had COVID last year and who's had chronic fatigue, brain fog, all kinds of other symptoms, is just feeling unwell. This is a huge structure. If you see that these are red blood cells, this is exactly what we're seeing in the vials. It's a folded up carbon tube. This is most likely, it can be from shedding. And this is a real concern. This is somebody who has an elevated D dimer, meaning their blood is clotting. If it is graphene, the pH of graphene is 1.3. It's an extremely acidic molecule that leads to this type of rouleau formation where the red blood cells no longer can carry oxygen. I found this in an unvaccinated person. This looks just like what Shimon showed in terms of these magnetic bead-like structures. What is this doing in a healthy person, middle-aged, who has no symptoms? My concern is a lot of these people are also asymptomatic. Most of the people that I'm doing live blood analysis on now, I'm finding structures in them. This is clearly not normal. And you can see how the red blood cells are abnormal around it. There might be some electromagnetic phenomenon that is affecting these people. Here again, you can see these huge structures. If you look at a red blood cell, this is hundreds of times larger. With this rouleau formation, it is affecting blood circulation. And these fibers back here, this is fibrin. This is causing blood clotting. Okay. And so right here, you've seen in Shimon's images exactly these kinds of strange things. So some of our colleagues are saying that these are, again, crystals and that they're thinking that this is fibrin. I don't think so. And I've talked to scientists who've done live blood analysis for 30 years. They say they have never, ever seen something like that in such a massive scale in people. Look at how large this is. These are all unvaccinated people. And this is a shedding phenomena. This is environmental poisoning. It's having effects. I've been a physician for over 22 years. I've never seen this amount of brain fog and dementia-like symptoms in 30-year-old people who had presumed COVID. And here, this is what's in their blood. These are huge carbon structures. And look at this blood. It's just clumping together. Unvaccinated people. And
Anna, how do you know this is caused by shedding or by environmental toxins?
We don't know where it's coming from. So our colleague Matt Taylor, and I've done a video with him, he found these structures in rainwater because it's sprayed in chemtrails. We have people who have analyzed the rainwater. It's clearly graphene carbon structures. We have it in food supplies. We've people who found it in even carbonated mineral water. We know that the mRNA injections are being given to the animals, so it might be in the food supply. But there are a lot of people who are saying that they have symptoms when they are around vaccinated people. Like I said, women who are just hemorrhaging and even postmenopausally, and it doesn't stop these extreme fatigue-like issues. And I think that a lot of people are complaining about symptoms that are around vaccinated people. Here's just another image. This is this rouleau formation, and you can see the amount of fibrin back here in somebody who has these kinds of graphene-like findings. So I think that I just want to very briefly say there are possibilities, for example, with EDTA that can dissolve hydrogel. It can take out graphene. It does take out metals. I use it IV. There are other modalities, for example, through the skin. Humic acid has been found to dissolve graphene. Nitric oxide is very important because the microbiome can help with detoxification. Vitamin C is a chelator of metals. NAC, glutathione, dimethylglycine. Peroxidases have been shown to dissolve these things. For example, that's in horseradish. Eat organic, have clean water, use ionic foot baths and sauna. And the reason why I'm saying this is while people are spending a lot of time discussing this, they're going to attack us, they're going to, discredit us. There are people that have ticking time bombs in their body. And what I'm saying is, in addition to everything else that they're doing with these vax injury protocols, also address these metals, also address the graphene, start getting microscopes and just saving people's lives.
That makes sense. We still don't know what exactly, we know what we're talking about. We're talking about structures that are probably devices, and we know this stuff doesn't belong in the vials, and it certainly doesn't belong in the human body. We don't know what it causes, apart from the fact that it probably causes thrombotic reactions, blood clotting. But we need help. We need more. We need to investigate. We need to look into this much deeper. But I think this is an excellent first step to get this out to the broader public, to as many people as possible, so that hopefully people will join us and do their own research, experts who might be helpful to us. And I didn't know, Anna, that this stuff was even found that these devices were found in animals and in water and in food, apparently. Are you sure about that?
It's in the patents, by the way. They state. It is. I can show you right now if you want to. We can look at, or I can email it after. Yeah.
It's extremely concerning.
It can be put in clothing too. It can be put in anything. These are nano. Mm-hmm. They're AI nano biological nanoparasites basically, and they have ionic charges, and they can penetrate surfaces and infect people. I mean, it's in the patents. They explain what they're doing. They even specifically mention that they're going to put it in combat wear and coat military equipment. They say they'll coat workout like gyms down with it too. So.
People are never going to understand this unless you understand that we are right now in World War III, and that's not conspiracy, and that humanity has been attacked, and that we need to really wake up from this reality. Oh, it's just about science, and maybe we need to publish the next paper. Well, the entire medical literature, 70% of that is fraud according to their own editors. And I just think right now it's about, we need to save lives by getting the information out. I agree with you, Reiner. If people don't want to work with us or dismiss us, I don't even care. I don't want to spend a minute on them. It's like, let me get to the people that can hear me and that maybe have vaccine remorse and say, hey, try these things to maybe save your life. Get away from your cell phone. Get away from Wi-Fi, because if there is a possibility that this is related, until people figure it out, just be safe so that we don't have more died suddenly 20-year-olds.
Sure. So, Anna, I don't know if you're in touch with the Truth for Health Foundation. I think Sasha and Hedley and me have worked with Dr. Lee Vliet, and she's worked up. She's brilliant. She's working with anyone who's got insight into recovery, vaccine recovery. So if you're in touch, great. If not, you'd be welcome with open arms. She has a platform and developing protocols, and because it's not subject to rigorous, placebo-controlled trials, it has to be done by, people with good heart, good heads, and a strong rationale. So that sounded like where your list came from. So would you like an introduction, or do you already know her?
Yes, I would love to. Thank you.
Great.
Please, please.
Yeah, I will do that.
This is our most important task, to connect with everyone who can help. And that's what we're doing.
If I could just touch on the device thing, it's just like we need protocols for dismantling bombs, biological, electronic bombs that are planted inside of people. And we need to heal people of this.
We first need to find out what this is. What we do know now is this doesn't belong in the vials, it doesn't belong in the human body, and it looks like we're dealing with devices. It doesn't just look it. As you showed us, Karen, these things are devices. Mm-hmm. If this changes everything, and I believe it does, in a court of law, that would be extremely helpful. But what I do think is that if people see this, both the unvaccinated and the vaccinated, it's going to change their minds. It's going to at least make them ask questions. And if that is a good first step, then I'm happy with that. David.
Yes, I just wanted to make the point that some of the more wacky stuff in the video that I showed at the beginning, there are a group of people who need to be aware of that potential. And it's not the general population, it's not the people that are vaccinated, but it's the people that are looking at what this weapon does and what the scope of the problem may be. Because this could potentially be part of that spectrum. And so there is a subset of people who need to be aware of that may not be aware of it.
Those are the people who we're trying to reach. We're trying to reach as many people as possible, but that target group is probably the most important one, the one that you just mentioned. Because one thing is certain, as far as I'm concerned, and just like Mike, up until 2.5 years ago, if I had heard any of this, as I keep saying, I would have told them, take your pills, go see a doctor, you'll be fine. But in the meantime, I'm absolutely convinced that this is about life and death. It has never, ever been about health. Everything has been stood on its head. This is about life and death, and that is the most important message that we need to get out. And if once you understand this, and all of a sudden the things that we've seen today appear in a completely different light, because they may be used for killing us, as these devices and these so-called vaccines have done already. And even if you look at Steve Kirsch's estimate, I think is what you might want to call it, based on the VAERS data, 1.6 million people in the US alone died after getting these shots, after getting these devices maybe injected into their bodies. We'll see.
I think the thing is everything's been a lie. And if you read one of my Substacks, these are actually considered neuroweapons, biological neuroweapons. Mm-hmm. And for James Giordano, who works with 2 Congresses on neuroweapons and weapons of mass destruction, he even says it would be illegal for us to use this on the American population or human population unless we did it under a medical experimentation with a private entity where transparency wouldn't be needed. This is on James Giordano's website. It's in one of my Substacks. So this is truly a war against humanity, where they said, look, if we were to do this, it's completely illegal and we'd be charged with crimes against humanity. But if people agree to it under experimentation, and we didn't have to be transparent because of our partners, because of intellectual property, then we're not doing anything illegal. This is literally on his website.
Yeah. Interesting, Karen. I read yesterday, I think it was a Substack article, which I can find. You may know this story already. It turns out that the White House worked with a bunch of different branches of federal government, and to my great surprise, because I'd not thought about it, the group that was in charge of strategy and operations was not CDC or FDA or anything like that, but FEMA. And it is. I didn't know that until yesterday, and I didn't sleep very well, because it was this. FEMA have got nothing to do with public health, as you know, and yet there it is. There are memos, names, organizations, dates from the Trump White House. I'm not blaming him, but it's just basically they'd spent 20 years rehearsing pandemic preparedness plans through HHS, CDC, that sort of thing, FDA, and then in February, I think it was, or March, just literally, it seemed arbitrary and capricious. The entire responsibility was moved to a branch of federal administration that has no competence or experience in dealing with it, and the other groups were cut out. So I only learned this yesterday, and I wonder, it probably means that the military has always run it, and that all of the rules we thought applied, are probably in parentheses and are all suspended. So
yes, absolutely.
It's dreadful. Mike, I read it and I thought, they probably did it like this because no one with competence would be inside the administration and be able to say, you're doing it wrong. It's like the FEMA people wouldn't know what they should do. HHS was not technically involved and there's nothing to say about it. The regulators are probably told, because of the way this is coming at you, you just have to say yes, you're required to. So, it gets worse and worse, but I'll send that paper out, that Substack article, and please judge for yourselves, folk, is that what's happening? And if so, what the hell was behind it? But it could affect what we do.
And your suspicion, Mike, is confirmed by the fact that if you look at the exercises, the first one was Operation Dark Winter. That was a military exercise. Yeah. And if you look at the defense that Pfizer gave in the Brooke-Jackson trial, they said, we didn't have to adhere to any rules because we're under a special, I forget the name, under a special contractual obligation with the military, with the DOD. The only thing we needed to do is supply them with, 50 million prototypes or something. So it is definitely the military. It is what some people call the deep state. It's not all of the military, but the deep state that we're really looking at. We know that, but...
Pfizer's considered a covered person in that contract, and this is why the FDA approval is so important. In that same contract, I've read everything. In that same contract, I even have this highlighted. The DoD specifically says Pfizer and BioNTech are working with the FDA for FDA approval. Yeah. That is a separate procedure and is not under the DoD supervision. That's why the approval is so important. The shield is broken.
Yeah.
In their filings with GeoVax, the holding company for all the patents, they weren't supposed to even submit for FDA approval until June of 2025. They got a lot of pressure. Fauci freaked out and pushed for the FDA approval, and he broke the shield. And then they had to make sure that the people that were trying to win the battle for humanity were convinced that it never was broken.
Yes, I understand that. Friends, we have to come to a close unless there's some important additional remark that needs to be made. But I think we got the point across. We're dealing with really strange structures that probably are devices, and we're dealing with the military. We're dealing with something that is definitely dangerous for human beings. It shouldn't be in the human body. So, is there anything else that we need to add to this? This is a first step. I think we're going to have to do this again.
Reiner and Mike, thank you so much for even allowing us to present this again. This is so controversial. Many people don't even want to hear about it, don't want to listen. Thank you so much for allowing us on this platform, and thanks so much to Karen Kingston for her brilliant work. And obviously my colleagues, but I think it's so important to at least allow a dialogue, then you can see for yourself. Yes. Really appreciate it. Thank you.
Everything needs to come out, because if it doesn't, we won't have the full picture. We need the full picture in order to make the right choices. And I thank all of you, Anna, Shimon, Karen, David for staying up so late. And of course, Mike, I'm very grateful for you helping me. And this is the first time we've ever done this, but it's probably not the last time. So thank you very, very much.
Yeah, thank you. I add my salute of gratitude and thanks to all of you, working away individually and with others to uncover this monstrous crime and hopefully be able to throw handfuls of greasy ball bearings under the feet of perpetrators. I want to live to see that.
Yes. We will. So Thank you very much and have a great weekend.
Cheerio.
Bye-bye. Thank you all.
Cheers.
I'll be sending you documents. Thank you.
Okay.
Excellent. Take care. This was another session of the International Crimes Investigative Committee, and this time we were really talking about crimes. There's no other way but to call these things crimes. If the injectables, as Shimon calls them, the devices that are being injected into the human body, if this is what's happening, then we're definitely not talking about vaccines. We're talking about the human body being misused for a purpose that we it is probably too horrific to even think about it, but it is important for us to know that this is going on, and it is important for the people out there, and especially those who are thinking about getting the so-called vaccines and those who've gotten them already, to understand that this is something very, very dangerous, and this is a crime against humanity. Thank you very much.